Hello students, today I will tell you about the blood vascular system. Here is the question, this is the syllabus as per according to the National Education Policy. Now here is there are three components that blood. The first question is what is blood? Blood is a liquid connective tissue and it transports along with the vessels, therefore here is the vascular and it follows a pathway, that's why here is the system word used. The blood vascular system means the transport of a liquid inside to the vessels in a pathway of the body, that's why this system is known as blood vascular system. This blood vascular system is a lifeline system of the organisms because this system plays many important roles for governing all the physiological functioning of the organisms. Here is the syllabus, that is hematopoiesis, composition, function of the blood coagulation and brief account of the immunity, types of heart, origin and conduction of heartbeat and cardiac cycle. But in the present lecture, I will only tell you about the hematopoiesis. Hematopoiesis. Here is hematopoiesis means heme, this word coming from the iron. Heme means iron and poiesis means the formation of the blood. Now we will see just like this is hematopoiesis. Basically, hematopoiesis occurs in the bone marrow. Bone marrow means where is the bone marrow? If you talk about this is your humerus, and in the case of the bone of thigh, that is femur. In case of the long bones, the soft spongy tissues is present, and that soft spongy tissues is known as the bone marrow. And inside to this bone marrow, the formation of the blood occurs. Basically, if we talk about in case of the blood, blood has 55%, approximately 55% is plasma, and rest part of blood is formed elements, 45%. Under to these formed elements, the RBC, red blood corpuscles, white blood corpuscles, and platelets exist. Here, if we talk about this one, and this plasma has the many proteins and other things are also exist. The question is how can you separate the plasma? So at first, so what you have done, you bleed to the animals, and then and you need some anticoagulants in your vials. You pour some anticoagulants in your vials, then take the blood in that vials, and everything would be in the ice. Then you should go to the refrigerated centrifuge. In refrigerated centrifuge, in a certain RPM value, you can just do centrifuge to that part. So the supernatant part will comes in the form of plasma and the formed elements inside. So now with the help of Hamilton syringe or micropipette, you can separate to that plasma, and the rest part is the formed elements that are your the RBC, WBC and platelets. If you uh, but whenever you are working on the serum, so in case of serum, what you do, you just to take the blood and then coagulate to that blood in the air dry conditions, then to centrifuge that blood in 3000 rpm, and the supernatant contents will get as the serum. So in case the serum is also an important part of this one. But here is our target is that these are the precursor cells, that is the hemocytoblast, hematocytoblast. This hemocytoblast blast means these are the stem cells. Now the question is, what is stem cells? So stem cells are those cells which can differentiate to any types of the cells. But when these stem cells become progenitor cells, so the basic difference between progenitor cell and stem cells is the fate is determined in case of progenitor cells, but fate is not determined in case of the stem cells. That means the stem cells can differentiate any types of the cells. On the other hand, the progenitor cells can differentiate to their specific targets. Here you will see this is the hemocytoblast, and this hemocytoblast has again subdivided into another set of the stem cells with the help of some mitogens such as the proerythroblast, myoblast, lymphoblast, monoblast, and megakaryoblast. If you talk about this protoethioblast, then if this proetoblast undergoes to the some certain mitotic divisions, then it becomes the polychromatic erythroblast, then the erythrocytes will becomes. Here will be happening there is some proerythrocytes because this diagram I have taken through the Creative Commons license. So here this proerythroblast has been changed into the proerythrocytes, then it changed into normoblast. In case of the normoblast conditions, the nucleus is disintegrated in the mammalian RBC and RBC will become the enucleated. After that, these proerythrocytes will differentiate into the mature formation of these erythrocytes. These are known as the red blood corpuscles. On the other hand, if you are talking about in case of this myoblast, here is this myoblast is undergoes to the some mitotic divisions, then it becomes the progranulocytes, and these progranulocytes will subdivided into basophils, eosinophils, and neutrophils. These are the granule-like structures are present in these cells. That's why these cells are known as granulocytes. The question is why these are known as basophils, because the basic dye stains to this one. That's why it is basophils, and the acidic dye stain to this, then it becomes eosinophils and neutrophils. Both the composition of the basic and as the acidic dye act on it. That's why these are the neutrophils. Here is an important question: is that neutrophil is the first cell which resides at the site of infection. You have to note down it for your competitive examination. In case of the lymphoblast, other hand is the lymphoblast. This lymphoblast certain undergoes to the mitotic divisions with the help of some mitogens or interleukins. Then it becomes the lymphocytes. Then ultimately these lymphocytes will become either differentiated. The maturation of these lymphocytes will determine its nature. If these lymphocytes will mature in the thymus gland, then they become the T lymphocytes. If other than the thymus gland, these are mature, then they become the B lymphocytes. Basically, this T lymphocyte is responsible for the cytotoxic toxicity, cell-mediated cytotoxic toxicity, and the B lymphocytes, whenever the antigen comes in the bodies, the B lymphocytes will differentiate into the plasma cells, then the plasma cells will produce the antibodies for the specific antigens. That is known as the humoral immune response. In here is the monoblast. These are the monoblast. These are also the stem cells of these monocytes. Then again in the certain mitotic divisions occurs in these monoblast cells, then is differentiated into the monocytes. This monocyte ultimately this monocyte differentiate into the macrophage cells. Also, it's showing the here. This shows the phagocytosis. These are the agranulocyte. Here is the granule-like structures are not present, and the granulocytes and agranulocytes will produce the leukocytes, and these leukocytes will move as white blood cells. These are the defense mechanism. These cells plays an important role in the defense mechanism or the immune mechanism of the organisms. These are the megakaryoblast. These megakaryoblast then subdivided into the megakaryocytes, and that with the help of some mitotic divisions or mitogens, ultimately these megakaryocytes will produce the platelets, and these platelets will play an important role in the blood clotting mechanisms. So my dear students, here are some specific points that in this lecture I summarize this lecture. When you have to think about, after listening to this lecture, you should think about that what is blood, and where is the formation of blood occurs, and what are the stem cells, and what are the progenitor cells, what is the basic difference between stem cells and progenitor cells, and uh, next is that the composition of the blood, the plasma, what is plasma, how we will separate the plasma from the blood, and what is serum, how we can separate the serum from the blood, as well as the formed elements, and what are the formed elements, what is why RBC is enucleated. The RBC is enucleated because this RBC transport the oxygen, and so the nature supports for the transportation of the oxygen process. uh, that's why in normoblast conditions, this RBC becomes enucleated, and the agranulocytes and granulocytes are the parts of leukocytes and the platelets. The whole lectures is basically uh, the focus the understanding of the blood. Uh, for any questions you have, you can my email ID is here. You can email me. Email me. I will just solve your questions as soon as possible. Thank you very much.